Hello, I am Henry Borbach, host of Reef Exchanges, the podcast brought to you by the Reef Resilience Network. For over 20 years, the network has been a global leader in training marine managers and other practitioners to protect, restore, and maintain coral reefs. We do this by turning scientific information and in the water experience into operational knowledge, tools, and guidance. You can learn more about all that we offer at reefresilience.org. So, without any further ado, let's dive on in. On this episode, I'm joined by my colleague Anik Cross, Resilience Science Lead for Global Oceans at the Nature Conservancy.
Hi, everybody. Nice to be back on the podcast.
And we're both joined by Dr. Alwan Hilkema, Associate Professor for Coastal Ecosystem Restoration at Van Hal Larenstein University of Applied Sciences and Wageningen University. We're going to be talking about Alwan's recent paper on herbivory restoration, a summary of which you can access at reefresilience.org. But before we get too far along here, Alwan, will you please introduce yourself and tell us a bit about your background?
Yeah, sure. Happy. And thanks for inviting me for the podcast. So my name is Alvin Hoekema. I work for Van Hal Larenstein, University of Applied Science, and Wageningen University. And I work from Saba, a small Dutch Caribbean island, where I do applied research into how we can restore our coral reefs. And my research focuses on grazer restoration. So at the moment, we have so much algae on our reefs that corals are barely able to recruit and to recover from other disturbances because there's not enough grazing. So my research focuses really on that part.
Thanks so much, Alwyn, for this great introduction. As we mentioned earlier, the Reef Resilience Network, just did a summary on a great paper that you co-wrote, co-authored. Restoration of herbivory on Caribbean coral reefs. Are fish, urchin, or crabs the solution? But before we jump in talking about that, could you touched on it a little bit, but could you explain to people what's happening on the Caribbean reef and why there is so much algae? Why are you finding so much algae on it?
Right. Yeah, that is a combination of causes. There's eutrophication, so there's too much nutrients in the water that sort of fuel the algae to grow. Also, we have less grazers than we used to have in the Caribbean. There's overfishing of herbivorous fish for decades already. And there's also a couple major die-offs along the sea urchins that are also important herbivores. And then another sea urchin species has also been overfished. So that the combination of these things resulted in faster growing algae, less grazers. And then there's, of course, a variety of causes that also resulted in degradation of the corals. So that means that coral cover decreased, giving more space for algae to grow. So that means even more algae with less grazers. They simply cannot keep up.
And so do you think that with increasing the number of grazers on the reef, you would be able to help to increase the resilience of those reefs?
I think so, yes. Of course, there's multiple things that need to happen. For example, we also have to look into those nutrients to reduce those if possible. But if pilot studies have shown that if we're able to increase the grazing pressure, then algae are reduced and coral recruitment can actually increase.
And tell me a little bit. So, what is your research about? So, which species are you focusing on? In your paper, you talk about fish, urchins, and crab. And I know you described very different approach for this. Could you start with explaining a little bit about what you're trying to do with fish or what you looked at?
Right. So we wrote this paper with a group of experts. And my research so far, my contribution to the paper is mostly about the sea urchins.
Okay.
And we also started looking into crabs recently, but we didn't look into increasing fish biomass. It's very difficult, to be honest, to increase the the fish biomass on the reef by culturing them. The easiest way to do that is to prevent them from being fished.
So for fish, we're thinking more protecting them from being fished. And so what is your research saying about urchins? Can they be brought onto the reef?
Yeah, absolutely. It's a species that is challenging to culture, but it's absolutely possible, and that also has been done already. So we developed a method to culture them. A partner university in Florida also developed a method, and they can be raised in captivity. And the biggest bottleneck at the moment is doing that in numbers that actually make an impact, and keeping them on the reef. Because as soon as you put them on the reefs, there's a lot of predators out there that actually miss the urchins as part of their diet, and they immediately attack the restocked urchins. So that's another bottleneck that we're working on.
What was the cause of the die-offs initially? Does that still apply another pressure as you're trying to restore the populations?
Yeah, that's a good question. So there have been 2 die-offs for specifically the diadema sea urchins. So diadema are the black sea urchins with the very long spines that most people actually hate. Most beachgoers hate because they can step on them. But they're very important for the reef. And there was one die-off in 1983, 1984. The cause for that die-off has never been confirmed. And there was a recent one in 2022. And when that happened, there was immediately a lot of action to find out, like, okay, what is the cause? We have to know this time. We cannot let it pass. So we, with a group of researchers, we collected samples all over the Caribbean, healthy and affected sea urchins, and then we were able to identify the causative agent, which was a sceticociliate, a small unicellular organism that specifically affected this species of sea urchins, nothing else.
And do we know where it came from?
No, we don't know. We did find out, or other researchers found out, that it is still in the Caribbean. It can be found on the bentos, but it does not seem to affect sea urchins in its normal state.
Right.
But this spring, again, there were a couple areas where there was small localized die-offs. So it might be something that maybe came from somewhere else, but is now local, abundant in the Caribbean, and might be triggered by certain environmental conditions to proliferate again. So this is something that's pretty concerning, actually, because we don't know yet, but it might stay a continuous risk for natural and restocked diadema populations.
You mentioned that you needed a certain density of urchins to start making a difference. Do you know, can you explain to the listeners a little bit like what it would take to start seeing those densities again with the type of rearing that you're doing in the labs?
Sure, yeah. Depending on where you are and how deep you are, how much nutrients they are, that all depends. The growth rate of the algae, of course. How many other benthic organisms also have an effect. But it's thought that between 1 and 4 adult urchins per square meter can actually make an impact on the reef, depending also how rigorous the reef is. But to get 1 to 4 adults, you need to restock many more juveniles to actually make it to that many adults. So yeah, that maybe gives an idea that to restore not too big of a reef patch, you already need like a couple thousand of individuals.
Wow. For how much area? You said a reef patch, but what's the area more or less? Maybe like 10 meters square or something like that.
Exactly. Yeah, something like that.
Yeah. Okay. That's a lot.
Well, yeah, I think with 10 times 10 meters.
Yeah.
So for 10 meters square, you would more need something like 100 to 400 urchins.
Okay.
That, yeah, adults. But then, yeah, to juveniles, yeah, around 1,000, I think. Yeah.
1,000 juveniles.
So that's a lot. Yeah, you're right.
Do you think that this is something that people who are managing, who are this could be useful for areas like marine protected areas, or people who have hotels, for example, who'd be interested in getting their reef more resilient to have more fish? Or I mean, it seems like a lot, and the Caribbean reef is big. So
Yeah,
how do you envision using something like that? Is it small scale in some critical areas, or are you thinking about ways of scaling this effort up?
both. So
Okay.
one thing is scaling it up. At the moment, we can produce a couple hundred diadema per culture run, but there's definitely potential. We're now testing a new method in which we can keep around 100,000 larvae in a culture vessel. So that's a good step. And yes, for the critical areas, for example, the areas where we are trying to outplant corals, to give those corals like a chance, a bigger
Right.
chance of survival is of course very useful. And then third, the good thing about diadema and other sea urchins is that they create a habitat that is very much liked by their own offspring, by the juveniles. So one of the issues, and I did a lot of research into the why the sea urchin species not recovering naturally, and it's a bunch of different factors depending on the location. But one of the things that is clear is that there's a lot of places with actually quite good settlement. So larval influx is not really the problem. It's
Right.
mostly the conditions on the reef that are unsuitable, and those change as soon as you have like some adults there that graze away the macroalgae and yeah, get a more friendly habitat for the juveniles. So my hopes are also that if we restore them in certain places, that will also trigger the natural recovery. So then we don't have to do all those thousands and thousands by ourselves.
That reminds me of another case study we have on our website from some work that the Nature Conservancy was involved in Hawaii in Kaneohe Bay, where they were doing mechanical removal of algae, of invasive algae there, but then they've been able to maintain uh they've maintained that work what just with urchins. Like they no longer do any mechanical removal of algae, but they are doing that kind of urchin based
Yeah.
um right, or I guess sort of restoration maintenance. Is that sort of when you're talking about like the initial reef health, is that sort of what you're talking about?
Uh, yeah, that is mostly where I'm talking about, but also that once you manage to establish a couple small populations, they will have a positive effect on the local settlements, but also produce gametes themselves. So they might seed out to spill over to different other places. And the time in the water for the larvae is very long. That's also one of the challenges during cultivation. It's up to 60 days. So they can spread very far.
Alan, you're on the research side of this work. And I'm just wondering, let's say, for example, somebody is interested uh, on another island, somewhere else in the Caribbean, in applying some of what you found for their marine protected area, for example. Are there plans on turning what your research is doing to rear these urchins and then put them out there? Is there going to be a way to collaborate with others? Is it going to go commercial? Like, how do you anticipate this happening, and how do you anticipate sharing all that knowledge and like practical know-how outside of your research?
Yeah, yeah. We try to share as much as we can. We have been involved in workshops, diadema restoration workshops, or reef restoration workshops. We also regularly have people from other organizations that come here for training. Um, so I see it as we are working on a sort of an approach that can be applied on other islands where we're just not there yet with the diadema and the sea urchins, but also not too far from yeah, ready for application. And at this moment, I think it's great if there's other places that have the capacity, because it does require quite some capacity to culture the sea urchins. But that we do tests in different other parts of the Caribbean, because every place is different. What is a bottleneck on Saba does not necessarily have to be a bottleneck in the Greater Antilles or in another place. Yeah.
How complicated is your setup and how expensive is it? Is it something like, for example, if the French government in Guadeloupe decide to do something similar, they could set it up, or how complicated is it?
Yeah, it's not super complicated, but you need like dedicated staff and funding to do it. Like definitely
Right.
a lab with where you can regulate the temperature, have access to good quality seawater, those kind of things.
Right. And know-how, right? And the skills to
Absolutely.
maintain all of that.
Yeah, and it takes a bit more than you think, because the larvae, they eat microalgae.
Right. So you
And
got
you
to
have
rear
to,
that.
yeah, exactly. You have to rear the algae as well, which is a whole thing by itself. Yeah.
Yeah, I live in near Monterey, and so the Moss Landing Marine Lab has a whole setup as well for urchins, and I didn't realize that it was exactly the same process for cold water urchins as it seems to be for the Caribbean urchins, actually.
It is pretty similar, but what differs a lot per urchin species is the pelagic, the duration of the larval phase, so the time they spend in the water column.
Right.
So for some urchin species, it's close to 2 weeks or even 10 days. For the didyma sea urchins, it's 6 to 8 weeks. So
Oh,
it's pretty long, and that means that they're pretty sensitive as a larvae. So all the time you have to make sure that all conditions are right and you feed them the right microalgae. So that makes it challenging. And we're also looking into culturing trypnosis sea urchins.
yeah.
Those are
Yeah.
the West Indian sea eggs, the big white urchins. For a couple reasons, so that is the species that has been historically overfished. So it was more
Right.
abundant than it is at the moment. It gets pretty big and has a high algae consumption, and has been shown already that it eats macroalgae very well. We do some experiments to see if it is complementary to didyma. The good thing, it's not affected by the die-offs, so that is a positive thing. And also the rearing time of the larvae is around 17 days, so
Right.
considerably shorter than the didyma.
The problem is the eggs are considered a delicacy for many people, right?
Yeah, it's very island dependent, but
Oh,
I can
really?
imagine that, yeah, like here on Saba, there's barely any interest for most of the Dutch islands. It's not really a thing. But if you
Okay.
go to the French islands, for
Oh,
example, then
yeah.
yeah, they really like them there. So that might make it harder. Like you really have to co-manage them together with the fishers to agree on certain areas where you restock, and then that they don't just. But I think it can be combined. Yeah, we're actually doing a
Right.
project because the king crab, which is another important herbivore, they're also a delicacy in some parts of the Caribbean. So yeah, I think the didyma might be more the exception, and for the other species, you have to work with the local communities to make sure that they don't fish away what you just put in there. But it's in their long-term interest as well, of course. If the reefs
Right.
are healthier and there's more urchins, and they could fill on the fish on the spillover in neighboring areas.
You talked about the crab. Can you tell us a little bit about your research on the crabs, and are they similar herbivores as the urchins? Do they have the same role? Is it more complicated? What's the story with the crabs?
Sure. Yeah, happy to talk a bit more about that. Most of that research has been done by Mark Butler and Jason Spadaro, among others. We are doing a little bit research on the crabs now, and also we want to see how their interactions are with other herbivores. But regarding the culture, they're actually much easier to culture. They have a very short larval phase, and they don't eat during those couple days. So it's mostly having a good water quality, some settlement substrate, and it's relatively easy compared to the urchins to culture like a couple thousand.
Right.
The bottleneck for the crabs is mostly how do you keep them on a certain reef area? They tend to disperse a lot because they're very mobile animals. And
Right.
one of the concerns is also that they're pretty big. They can become really enormous, and they might not only eat algae. They're very good algae eaters. I was very impressed by how much algae they eat and how much they love it. But they might eat a couple other things as well. So that's still a research priority before this species can be cultured and restocked all over the Caribbean.
It used to be on the Caribbean. This is a native species to most
Oh,
Caribbean islands,
yeah,
or it's
absolutely. Yeah, I would only work with native species and
okay,
just
yeah.
either restoring their density to more historical values, as is the case with, for example, the diadema and the CX, or enhancing their densities, uh, to get some ecological benefits out of it.
So what else would the crabs eat than that you were a little worried about?
They might eat other invertebrates, maybe
Okay.
some sponges. Yeah, it's just like an open question. Like
Right.
so that's one of the things we're looking into now. But in general, it has been shown that they're really good grazers, and I think for the corals, they're actually even safer than the urchins. Like because also the urchins, if the densities are too high, then they will also start overgrazing the corals and can in the end even have a negative effect. But the
Right.
densities we have at the moment are very, very far away from that. So I'm not concerned about that at all. But the crabs are even more safe because they really have to sort of pick their algae. They graze in a different way. So they're not interested in the corals at all.
Owen, when you work or when you talk about your work in the Caribbean with just regular people, right? Your stakeholder, your fisherman, your tourism operator, etc. How do they receive this information? Like, are they excited? Are they worried? How is this work perceived?
They're generally very positive about what we do. Yeah. So yeah, it's something that doesn't harm them. Like I'm not telling them that they should do less dives on the reefs, something like that. I do sometimes tell them to fish less, especially on the herbivorous fish, because that's one thing, of course. I mean, if we try to increase grazing with the urchins or with the crabs, it is also important to protect the herbivorous fish because you cannot like it's so much effort to restore a couple urchins, and in the same way the parrot fish are fished away, then it doesn't make sense.
Well, thanks a lot. Henry, you you have a last question.
What one final question that I like to ask folks that are on these episodes, Alwyn, is for people that are listening, what advice would you have for a marine manager who's maybe read your paper and listened to this podcast and are looking to improve the management of herbivores in their area?
Right. Yeah. So I would say first step is protect what's there, right? Protect the herbivorous fish, protect the urchins, because in some places the urchins are actually removed, for example, in front of resorts. Next step is find out more about the capacity for natural recovery. So you can do that with settlement collectors. We develop those. So you can get a feeling of which locations might be very successful. If we manage to get a couple urchins there, then nature might do the rest. And then if you have the capacity, then culturing them on different places would be super interesting, and I think we can learn a lot from that. But you do need dedicated staff and a lab to do it.
Yeah, that's a theme that comes up often is the need for funding and capacity.
Yeah,
But
yeah, I know it's a thing. It's also it's one of the things I did a lot of ecological studies, looking at the effect of artificial reefs and looking at natural recovery of the diadema. And then I figured out, okay, we cannot really do this without being able to increase the densities, and for that you need like an aquaculture facility. So that's one of the challenges, I think, to go forward with this in different places of the Caribbean. But on the other hand, there is already a lot of labs, so maybe collaborations with local research labs can actually bridge this gap.
And I came away with a very interesting point that you made very earlier in our conversation, was that bringing these herbivores like urchins along with a coral restoration program could be really beneficial for the restoration program. And I think at the moment there's more money in these coral reef restoration projects. There's a lot of interest there, and I think combining this for me would be something that I think could really elevate this efforts to bring in herbivores on the reef. So to me, that what you mentioned there was really interesting.
Yeah, absolutely. We call it tandem restoration. So restoring the grazers and the corals at the same time.
Right.
And it has been shown that it is beneficial for the survival and the growth rates of the corals. So yeah, while doing that, even a small scale grazer restoration can actually have an impact by increasing the survival of the coral outplants. So that's a good first step, I think.
Perfect. I think that's a great point to end on, the tandem restoration and corals working together with herbivores and managers and scientists and the local communities and all of this together. And I think that is the picture of the resilient reef work that we're always imagining together. So thank you so much, Alwyn, for joining us. Anik, of course, it's always a pleasure. And thank you to all of our listeners as well. As usual, you'll be able to find the show notes for this episode and other episodes as well, including other resources like webinars, toolkits, and online courses on our website at reefresilience.org. And again, you can look up our recent article summary of Alwyn's paper there. And if you aren't already a member, please do join the network to receive notifications of new resources that we're always pushing out, including the next episode of Reef Exchanges. And we'll see you then.